So we know that the nucleus contains the genome, and the genome is defined as all of the cell's DNA and genes. When this gene is switched on, the enzyme known as RNA polymerase and the sigma factor in it attaches to the start of the gene. It moves along the DNA, and as a result, it exposes the bases and breaks the hydrogen bonds between them. So it breaks the 2 hydrogen bonds between adenine and thymine, and the 3 hydrogen bonds between cytosine and guanine. A strand of mRNA forms, which is exactly complementary and specific to the exposed bases, and it uses the DNA strand as a template for it. However, instead of thymine, mRNA has uracil, but it is still complementary to adenine. Phosphodiester bonds begin to form between the mRNA molecules. Before this strand of mRNA can be used as a template to code for proteins and amino acids, it needs to be processed. This processing can be called and known as splicing. It occurs when the enzyme known as the spliceosome cuts out the introns from the strand, leaving only the exons. They are cleaved at the 5' end of the new sequence as the exons are ligated together. The introns are also known as junk DNA, and they are non-coding sections of the DNA, and they amount for 98% of our DNA, whereas exons only code for 2%. Then the mRNA moves out of the nuclear pores and into the cytoplasm, where it comes into contact with the ribosomes. The sites in the ribosomes bind to the mRNA, and the ribosome reads the code to produce a chain of 20 different amino acids. This mRNA code is read in codons, which means it's read in 3 bases at a time by tRNA, which is transfer RNA. As the codons are read, the tRNA is complementary and specific, just how mRNA was when it used DNA as a template, but now tRNA is using the mRNA strand as a template. As a result, it delivers the corresponding amino acid, which is known as anticodons. The codons can also be known as the degenerate code, and the tRNA then carries the amino acids. As a result, the peptide bonds begin to form between amino acids. Ribosomes catalyze the formation of these peptide bonds. Peptidyl transferase is the part of this catalyzation. The chain of amino acids then folds into a complex 3D shape, forming the protein structure. The sequence of amino acids is known as a primary structure, which then leads on to the secondary, tertiary, and quaternary structure of proteins. And that's how protein synthesis occurs.